Hello, my name is Nuša Šavor, and today I'll be talking about the immunology of SARS-CoV-2. So, SARS-CoV-2 is a coronavirus of the genus beta coronavirus from the species severe acute respiratory syndrome-related coronavirus. As its name suggests, it comes from a family called Coronaviridae, which get their name from the look these S proteins, these spike proteins, these red ones, they give them a crown-like or a corona-like look. There are 7 coronaviruses which infect humans, most of which cause common colds, and in the past we have had 2 more dangerous and more deadly, more transmissive viruses, namely the SARS-CoV and MERS-CoV. SARS-CoV-2, as its name suggests, is genetically really similar to SARS-CoV. Other coronaviruses mainly infect birds and other mammals. So SARS-CoV-2 is a single-stranded positive sense RNA virus, and we'll get into what positive sense means a little bit later. It is transmitted through respiratory droplets and causes a disease we know as COVID-19. COVID-19 can range from asymptomatic to death, and some common symptoms are cough, fatigue, fever, muscle aches, and headaches, including a sore throat. Those are some of the more mild symptoms, although in more severe cases, it can lead to acute respiratory distress syndrome and pneumonia. The general anatomy is the RNA is situated inside a nucleocapsid protein. It's enveloped in a nucleocapsid protein, or the N protein, which protects the RNA from any hazardous impact the cell's environment might have on it, and it also aids in viral assembly, and it also could help it to fuse with the membrane of the host. The RNA and the nucleocapsid protein surrounding it are wrapped in a lipid bilayer membrane, on top of which we find multiple structural proteins, such as the M protein or membrane protein, the E protein or envelope protein, which aid in viral assembly and also viral exit, as well as the spike S protein, or perhaps the most important and most known protein to us, the protein which allows it to enter our cells. Another protein worth mentioning is hemagglutinin enterase, which could also aid in entry. So the spike S protein of the SARS-CoV-2 virus is a protein made out of 3 chains, which you can see right here, that it is heavily glycosylized, which means it has a lot of polysaccharides attached to it. These little squares and triangles and and circles, which is one reason why it is so good at evading our immune system. We here divide into 2 regions, the S1 and S2 region, which are important for the process I'll be describing. The SARS-CoV-2 virus also uses a number of host cell proteases or enzymes, which will transform the spike S protein into a shape which has more energy and will connect to the receptor, will bind to the receptor a bit better. It uses a receptor called ACE2, or Angiotensin converting enzyme 2, a transmembrane protein, most predominantly found in our lungs, but also in our heart, kidneys, liver, and GI tract. The angiotensin converting enzyme 2 is an enzyme which converts a hormone called angiotensin 2 to a hormone called angiotensin 1 through 7. Angiotensin 2 is a hormone found mostly in higher quantities, higher concentration in those of old age who live sedentary lifestyles and have poor diets, and it causes vasoconstriction, which in turn raises blood pressure. Angiotensin 1 through 7 is a hormone which is in higher concentrations in those of young age who live active, healthy lifestyles, and it lowers blood pressure through vasodilation. Both of these hormones are typically found in every human. It just depends on the ratio. In older people, angiotensin 2 is more predominant. And angiotensin 2 is a really important hormone because it brings necessary blood flow to the kidneys. It regulates if the kidneys get enough blood flow. Both of the hormones regulate if the kidneys get enough blood flow, and they're really, really important. So once the S1 domain connects to, first the S1 domain is worked on by these host cell proteases, and then it connects to the ACE2 receptor, it binds to the receptor. And once it is bound to the receptor, after being cleaved, it is bound to the receptor, and then we get more of those proteases working on the S protein, on the spike protein, allowing this S2 domain here to get lower, closer to the membrane of the host, and you can't see it right here, but it gets so close that it uses these two, like you can see the pink area and the blue area here. It uses it to connect to the host cell membrane and to fuse. Once it fuses with the cell's membrane, it lowers its RNA inside, and the RNA is then replicated and packaged, and we get new viruses. So it uses our free-floating nucleotides, our ribosomes, as well as our Golgi apparatus and our endoplasmatic reticulum. This is where the positive sense RNA comes into play. It makes this process a bit simpler for the virus. And how it goes on is this way. So the first 20,000 bases of the RNA are translated and transcribed into a RNA-dependent RNA replicase, which kind of functions as ribosome. So it takes that positive sense RNA and it feeds it through itself, and it pops out a negative sense RNA, which can either be it can go through the RNA dependent RNA replicase again and create small positive sense RNAs like these ones right here, which will be translated by our cells' ribosomes, and they'll make the lipid bilayer membrane, as well as the proteins which come on the lipid bilayer membrane. I'm sorry. And it can also be completely fed through the RNA replicase to make a complete positive sense RNA, which will be the genetic material for a new virus. So what would happen if I was exposed to a virus right now, to SARS-CoV-2? So the first thing that would protect me is my anatomy, my physical barriers. So my skin, the oils on my skin, which are technically the chemical barrier, but my skin, my nails, for example, my hair, and the chemical barriers would come next. So those are, as I've said, maybe the oils on my skin, the saliva in my mouth, my tears in my eyes, mucous membranes and the enzymes that come in my mouth, the enzymes that come in my GI tract, as well as bile, for example, the acidity of my stomach, the peristalsis, as well as the surfactant lining my respiratory tract. But if all that fails to protect me from the virus, if the load is high enough for the virus to be able to enter my body, it will enter through the nasal and oral cavity, and this is where the primary viral replication will happen, and the virus will go down my pharynx and my larynx to my trachea, to the bronchi, and lastly the alveoli. So I've explained how the virus enters the cell and how it replicates, and now I'll talk about the immune response, specifically to when the virus replicates itself inside a specific type of cell called the type 2 alveolar cell, or the surfactant producing cells of our alveoli. Surfactant is really important for our lung function because it contains a lot of enzymes that allow gas exchange, as well as it protects the alveoli from collapsing if there's any liquid coming into them. And if the surfactant is really thinned out, our alveoli will just, with one breath out, just stick together, and that will collapse them. So I'll go on to the next slide so you can get a better picture of the cells that are involved in this process. So inside our alveoli, we have those type 1 alveolar cells, which are those cells that perform gas exchange. They're really thin for the gases to be able to pass through, and those surfactant-producing cells are type 2 cells, which produce surfactant. We also have immune cells, like the dendritic cells, and also some macrophages. So the virus will be recognized when it enters our body, and all of these cells that you have here, the macrophages, monocytes, basophils, neutrophils, eosinophils, natural killer cells, and dendritic cells will have recognized them before, but we'll talk about what happens when the virus gets inside our lungs. So when it starts replicating inside those type 2 surfactant producing cells, they'll start releasing a lot of chemical signals that say, hey, we're being attacked, we need you to help us. And those chemical signals will recruit other immune cells. They will the first cells to the battle will be neutrophils. They are our most numerous immune cells. They create 50 to 60% of all immune cells circulating in our body at all times, and they'll come there and they'll also release a bunch of chemical signals recruiting other cells. We'll get macrophages, monocytes. Monocytes are just cells that will turn into macrophages, but they can circulate around our blood, our lymph and our blood, and get into the infected area where they will turn into macrophages. We'll get all of those natural killer cells, which are cells that kill infected cells. And the most and this all, like the sending of those chemical signals, and also these cells will release two hormones called heparin and histamine. And these hormones are really important because they will cause vasodilation. They will cause the blood vessels to get wider, to get more porous, allowing all of those chemical signals to pass through to the blood to be able to recruit those other immune cells. And they'll also, the porosity of the blood vessel right now will allow all of those immune cells that are coming to enter into the infected tissue. And we can see this the best when we hurt ourselves. For example, on our skin, you can see right now I've hurt myself on my pinky finger, and it's red. It's because of that inflammation and that pain we feel when we cut ourselves or when we scrape ourselves. That pain, that redness, is caused by the inflammation, by that widening of blood vessels. Okay, so the most important actor in this whole story are dendritic cells, these blue ones. They have nothing to do with the nervous system. They just are called dendritic cells because of these little legs, quote-unquote legs, which kind of look like the dendrites of a neuron. But these cells are also called antigen-presenting cells, or APCs. They will take a part, they're found in the tissues of our alveoli, and they'll take a part of that virus that came to infect the cells there. They'll take a part of it and they'll put it on their membrane. And once they put it on their membrane, they travel to our lymph nodes, and these are not the only lymph nodes. There are multiple lymph nodes in our body, but these are the ones I can show you. So they travel to our lymph nodes and they meet with a T cell, which is a lymphocyte, a type of cell produced in the lymph system. So the T cell, once it is presented with that antigen, will start proliferating. What proliferating means is it starts reproducing a lot, and the cells it creates are specific for, they carry that little antigen on them, and they can be either cytotoxic T cells or helper cells. Cytotoxic T cells will go into the lymph and blood, and they'll start looking for any cells that exhibit that part that were infected and exhibit that little antigen, because every cell that is infected, it also takes a little piece of a virus, in our case SARS-CoV-2, and puts it on them, letting the other cells know that they were infected. And those cytotoxic T cells will look for that. And once they find a cell that has been infected, they will put a lot of chemicals in it and cause apoptosis or programmed cell death. These cytotoxic T cells can turn into memory cells, which are just memory cytotoxic T cells, which will circulate in our body, so that next time we're exposed to that antigen, or in our case SARS-CoV-2, we are ready, we are prepared to attack it. The other type of T cell that is produced is a T helper cell. It has that antigen on itself, and it goes to a B cell, which is another type of lymphocyte. Those B cells are those cells that produce antibodies, and the T helper cell will show the antigen that it has on its membrane to a naive B cell, or a B cell that has never seen an antigen before. And that naive B cell will start proliferating a lot and into specific plasma cells that have specific antibodies for that antigen that the T helper cell presented to the naive B cell. And those plasma cells will circulate our blood and our lymph, releasing antibodies that can either stop the virus from binding by binding to the spike S protein, or once they're bound to the spike S protein, they are a signal to macrophages, which can recognize a part of that antibody and then engulf whatever the virus that antibody tagged. B cells also produce memory cells, which are just going to be those memory B cells that produce antibodies, and they'll remain in our bloodstream to protect us from acquiring that infection again. So both memory B cells and memory T cells, all kinds of T cells, so both T helper cells and cytotoxic T cells, will circulate in our body in lower quantities, but once they are triggered by the virus coming to our body again, they will start proliferation to create plasma cells and cytotoxic cells and helper cells. So we'll talk about what happens specifically in the lungs. You can see here what a healthy alveolus looks like, and once that blood vessel dilates, becomes more porous, it invites a lot of blood plasma in. So a lot of liquid seeps into it, and it starts filling the lungs. And a lot of those inflammatory signals can also escape from that part. If they're so strong, they escape from that area that is infected to other areas, and that is called a cytokine storm. When those chemical signals, also called cytokines and chemokines, they escape the infected area, they spread to the bloodstream, and they cause inflammation. They cause widespread inflammation, which can lead to sepsis or multiple organ failure. So the alveoli are getting filled with that liquid that seeps in through those porous blood vessels, and a lot of the cells, immune cells, are being recruited in. The virus is ravaging through those type 2 cells, and it also is now starting to infect type 1 cells. There are a lot of chemicals that are being released by those immune cells to target either infected cells to kill them, or to kill the virus, and that are really deadly for the healthy alveolar cells. And this is really bad. The alveolar cells are starting to die off, either because they were completely ravaged by the virus, or because of the harmful effects that our immune system, by trying to fight the virus, has on them. And liquid is starting to fill the alveoli. It's thinning out the surfactant, and just at one breath, what's going to happen is there's no more surfactant. There is no more gas exchange. We're having a really hard time breathing. The fluid our alveoli are full of fluid, and at one point, we're going to breathe out once, and it's just going to do this, and it's not going to come back. It's going to close, and this is when people need to be connected to ventilators. This is acute respiratory distress syndrome, and it is really, really, really bad. Of course, this is in really hard, really severe cases. This, you can just have moderate pneumonia at this stage, or if the liquid is kind of lower than this. But if it becomes really severe, what's going to happen is your lungs are going to start collapsing. Your alveoli are collapsing. You're having a really hard time breathing. Your lungs are filling with fluid. It's generally not a fun thing. And those cytokines, those chemical messengers, are traveling all throughout your body, causing inflammation in a bunch of other organs. So some other symptoms in other organs are in the lungs. You can get, dry cough, you have difficulty breathing, you have pneumonia. So, and that is all those like mild to moderate symptoms. But as I said, we have that severe symptoms, that severe acute respiratory syndrome or acute respiratory distress syndrome. That is when you really can't really need mechanical ventilation. All of those molecules, those chemical signals, and other chemicals are being released when the body is trying to fight off infection, can have a bad effect on the heart, causing heart attacks and decreased cardiac function and cardiac injury. Also, when all of that chemical messaging is being sent through our blood vessels, we can get a lot of plaque, because also the dead immune cells, the debris that's going on in that battle. You have a real battle going on, and a lot of that debris and those dead cells can create plaque, which can cause thrombosis and a heart attack. We also can get symptoms in our liver and our kidneys, because the liver, kidney, lung system with that angiotensin converting enzyme 2 and the angiotensin 2 and angiotensin 1 through 7 is really connected. So we can have acute kidney injury, and as well as hepatic damage, and a lot of accumulation of bad molecules in our liver. We can also get nausea and vomiting in our GI tract. We can feel really nauseated, and it can also cause diarrhea. We can also affect our nervous system with headaches, nausea. It's also been found that people get eye infections, such as conjunctivitis and conjunctival hyperemia, and some comorbidities. So medical conditions that can render you more susceptible to COVID-19, to severe COVID-19, are diabetes, hypertension. So those are people with hypertension are usually those who have an elevated expression of angiotensin 2, as well as cancer can be really bad, tuberculosis, and venous thromboembolism. So when you have a lot of when you have issues with thrombosis, so those little what causes heart attacks, blood clots. We have issues with blood clots. So generally, COVID-19 can be really, really disastrous for these people. And the only currently accepted medicine for COVID-19, which was authorized in the United States, is remdesivir. It is an antiviral drug that functions on the basis of inhibiting viral replication, or not inhibiting it per se, but making the virus, the new virus, defective. So it uses an analog to adenine, which is a nitrogen base used for production of protein. So it uses an analog to that, so something that looks like it, but is not quite it. So it forces the RNA replicase to pick up that analog, that that thing that looks like adenine instead of adenine and put it in its place. This makes the overall virus defective, and the translation will stop at some point, or even if it didn't stop, the virus is defective. So that is how it generally functions. Well, by now we have 4 authorized vaccines, the Pfizer, the Moderna, and the Johnson & Johnson and AstraZeneca vaccine. So with time, more drugs will probably emerge, and more vaccines will complete trials and get authorized. Now, mostly what treating this disease is focused on is stopping transmission, and also just stopping inflammation inside those who are infected. So here are my sources, and just to end all of this, this presentation was not meant to scare you to your bones and to make you terrified of SARS-CoV-2, but to make you more aware of what getting COVID-19, what becoming ill brings to some people. So how it can affect some people who have those comorbidities, or also now we've seen people end up with really severe symptoms, as we think, out of the blue. So just to make you more aware of the situation, so you can make decisions, you can make informed decisions. Thank you for watching.